The next 4 videos will be on the topic of glycogen metabolism. The first video, which is this video, will give an overview of glycogen, its structure, its functions, and how it relates to the skeletal muscle and liver. The second video will discuss glycogenesis, or the synthesis of glycogen, which occurs in the fed state, while the third video will tackle glycogenolysis, or the breakdown of glycogen, which occurs in the fasting state. The last video on this topic will cover the regulation of both glycogenesis and glycogenolysis. It will touch a bit on glycogen storage diseases, but I'll be leaving that as a reading assignment for you. Now, I hope you can still recall from another lecture the signaling cascade brought about by the stimulation of the G protein-coupled receptor by an extracellular ligand, which we also term as the first messenger. Now, as a review, GPCRs are the largest and most diverse group of membrane receptors in eukaryotes. Now, G proteins are specialized proteins with the ability to bind the nucleotides GTP, or Gold Tango Papa, and GDP, that is Golf Delta Papa. Now, the G proteins that associate with GPCRs are heterotrimeric, meaning they have 3 different subunits: an alpha subunit, a beta subunit, and a gamma subunit. Now, ligand binding to the GPCR causes a change in the receptor conformation that, in turn, binds and activates the G protein. Now, the active form of the G protein is then released from the surface of the receptor, dissociating into its alpha and beta-gamma subunits. Now, both subunits will then activate their specific effectors, causing the release of second messengers. Now, in this particular case, we have the adrenal medulla hormone epinephrine as our ligand, adenylyl cyclase as our effector enzyme, and cyclic AMP as our second messenger. Now, cyclic AMP in turn activates protein kinase, which in turn phosphorylates phosphorylase kinase, which in turn phosphorylates glycogen phosphorylase. Now, this series of phosphorylation reactions is what is termed as the phosphorylation cascade. Now, glycogen phosphorylase, of note, is a key enzyme in the breakdown of glycogen into its glucose 1-phosphate constituents. Now, take note of the numbers listed in the right column. Now, these represent the numbers of molecules of each member of the signaling pathway. Take note as well of the degree of increase in magnitude as we go down the signaling pathway. We call this phenomenon amplification. Now, this explains why, with only a small amount of epinephrine released by the adrenal medulla during the fight or flight response, a huge amount of glucose becomes readily available to fuel this fight or flight response. Now, from there, let me now segue into the basic characteristics of the glycogen molecule. So, glycogen is a large branched glucose polysaccharide whose glucosyl units or substituents are linked by alpha-1,4 glycosidic bonds, which I hope you still remember from the first biochemistry lecture. Now, alpha-1,6 glycosidic bonds occur every 8 to 10 residues in a glucosyl chain in order to allow branching in the glycogen molecule. Of note, despite the high number of glucosyl units, glycogen has only one glucosyl residue that has an anomeric carbon that is not engaged in the glycosidic bond. Now, that said glucosyl unit or residue can be found at the very beginning of the chain attached to a protein called glycogenin, which I'll be discussing in the glycogenesis video. Now, that being said, all the other ends of the chain in the glycogen molecule do not have free anomeric carbons and are thus non-reducing. Now, here we have a graphic representation of the glycogen molecule embodying the details I mentioned in the previous slide. Now, there is a biological reason why glycogen was designed to be branched. Now, this branched structure actually permits rapid degradation and rapid synthesis of glycogen by allowing enzymes to work on several chains simultaneously from the multiple non-reducing ends, thereby affecting the release of a large amount of glucose 1-phosphate molecules in a short time during glycogenolysis. Now, this slide features the alpha-1,4 and alpha-1,6 glycosidic bonds that the glucosyl units of glycogen engage in. You can pause this video so you can look at the illustration longer and be able to recall the difference between the alpha and beta configurations in this sense. Now, besides having many enzymes act simultaneously on multiple branches of the glycogen molecule, these enzymes are likewise in close proximity to the glycogen molecule itself, so that these enzymes can be called upon or easily utilized whenever the appropriate signal is given. Now, allow me to jump ahead a little bit. Here we have the summary of glycogenolysis or breakdown of glycogen. As will be discussed later on, glucose 1-phosphate must first be converted to glucose 6-phosphate for it to be channeled to major metabolic pathways. At this point, you should now understand that glycogen is the reservoir of glucosyl units. For that matter, glycogen can be tapped as an emergency source of glucose for ATP generation. Now, many tissues actually have their respective levels of glycogen in them, but the two major sites of glycogen storage are the liver and the skeletal muscle. The concentration of glycogen is higher in the liver than in muscle, that is 10% versus 2% by weight, but more glycogen is actually stored in skeletal muscle overall because of its much greater mass. Now, let's put these two organs side by side. Between the two, the enzyme glucose 6-phosphatase, marked by the red asterisk, is only in the liver. Well, in actuality, however, genes coding for this enzyme, though primarily expressed in the liver, is also expressed in the renal cortex and, to a lesser extent, in the beta cells of the pancreatic islets and intestinal mucosa, especially during times of starvation. Now, this enzyme functions by hydrolyzing glucose 6-phosphate, resulting in the creation of a phosphate group and free glucose. This free glucose is then exported from the cell via your glucose transporter membrane proteins or GLUTs. Now recall that this reaction completes the final step of gluconeogenesis and therefore plays a key role in the homeostatic regulation of blood glucose levels. Recall as well that the major site of gluconeogenesis is the liver, with a small amount also taking place in the kidney. Very little gluconeogenesis takes place in the brain, skeletal muscle, or heart muscle. Now, triggers for simultaneous activation of glycogenolysis and glycolysis generally depends on the type of tissue. In skeletal muscle, this occurs when ATP demands are high and when glucose 6-phosphate is being used rapidly in anaerobic glycolysis, as what happens during exercise. Now, in many other tissues or cell types, however, simultaneous activation of glycogenolysis and glycolysis occurs when flow of oxygenated blood to these tissues becomes restricted, with subsequent conversion into anaerobic metabolism. You will learn as well that glycogenolysis and gluconeogenesis in the liver have a common trigger or activator, the pancreatic hormone glucagon. Now, glucagon gets released from the alpha cells of the pancreas to activate these two metabolic pathways when dietary glucose in the blood decreases. Now, basically, the main goal of these two metabolic pathways is to maintain blood glucose at optimal levels. Now, just as a reminder, glucose 6-phosphatase is mainly present in the liver and kidneys. Now, before I get to the topic of glycogenesis, I would like to emphasize that glucose 1-phosphate must first be converted to glucose 6-phosphate for it to be channeled to major metabolic pathways. And the enzyme that catalyzes this is phosphoglucomutase. Now, phosphoglucomutase is an enzyme that transfers a phosphate group on an alpha D-glucose monomer from the 1' to the 6' position in the forward direction, or the 6' to the 1' position in the reverse direction. Now, more precisely, it facilitates the interconversion of glucose 1-phosphate and glucose 6-phosphate. Now, from the point of glucose 6-phosphate, the glucose molecule can now enter a variety of other metabolic pathways that include glycolysis and the pentose phosphate pathway. Now, let us lay down the groundwork for glycogen metabolism. Glycogen is both formed from and degraded to glucose 1-phosphate. The biosynthetic pathway, that is the synthesis of glycogen from glucose 1-phosphate units, is called glycogenesis. Now, this pathway is energy requiring, by the way. On the other hand, the degradative pathway, that is, the breakdown of glycogen back into the glucose 1-phosphate substituents, is called glycogenolysis. Of note, these two pathways, as you might have noted in the diagram, are separate and involve different sets of enzymes. And because of this separation, it is possible to activate one pathway while inhibiting the other. Now, as promised, I will be discussing glycogenesis in more detail in this video. But before we get to that, let us pay some attention to this intermediate here, uridine diphosphate glucose or UDP glucose. Now, UDP glucose is what's called an activated intermediate. An activated intermediate is a pathway intermediate which contains at least one high-energy phosphate bond for facilitating biochemical work. Now, these high-energy bonds, when cleaved, facilitate energy transfer that allows endergonic reactions to proceed in a forward direction. Now, since glycogenesis involves formation of glycosidic bonds, an inherently endergonic process, activated intermediates are necessary to participate in exergonic reactions that can be bioenergetically paired to the endergonic ones. Now, in the case of glycogenesis, the activated intermediate vital to the formation of glycogen is UDP glucose, as was pointed out in the previous slide. UDP glucose is formed from the cleavage of UTP to UMP and pyrophosphate. The phosphate of the UMP molecule then forms a phosphoanhydride bond with the phosphate of glucose 1-phosphate. The energy source for this bond formation is the cleavage of the pyrophosphate molecule. Now recall from the bioenergetics videos that the cleavage of a nucleoside triphosphate such as UTP to form pyrophosphate is a common synthetic strategy. Now, the free energy of pyrophosphate hydrolysis, which is negative 33.5 kilojoules per mole, can then be utilized together with the free energy of UTP hydrolysis to drive this otherwise endergonic reaction to completion. Now, the enzyme responsible for UDP glucose synthesis is UTP glucose 1-phosphate uridylyl transferase, sometimes called UDP glucose pyrophosphorylase. Now, other nucleoside triphosphate molecules involved in synthetic pathways is CTP, that is for lipid synthesis, and GTP for protein synthesis. The high-energy bonds of UTP, CTP, and GTP are all energetically equivalent to ATP. Now, here is the summary of the synthesis of UDP glucose. Additively, the change in free energy amounts to negative 33.5 kilojoules per mole. UDP glucose now has the high energy bond. When this high energy bond breaks, it would release enough energy to create a glycosidic bond between the glucose 1-phosphate unit and the end of the growing glycogen chain. The anomeric carbon of each glucosyl residue is attached in an alpha-1,4 glycosidic bond to the hydroxyl on carbon 4 of the terminal glucosyl residue of the glycogen chain. Again, to reiterate, the UDP glucose is a high-energy compound that can donate glucosyl units to the growing polyglucosyl chain of glycogen. To lengthen the chains, glucosyl residues are added from UDP glucose to the non-reducing ends of the chain by glycogen synthase, which we'll get to in a bit. Now, glycogen synthase utilizes the energy released by the cleavage of UDP glucose to UDP and glucose, which is an exergonic reaction, to form the glycosidic bond, which is an endergonic reaction. Thus, for every molecule of glucose 1-phosphate that is incorporated into glycogen, one molecule of UTP is hydrolyzed. UTP is then replenished via nucleoside diphosphokinesis. Now here is the UTP replenishing reaction. Since it is only involving passing around, so to say, of the phosphate bonds, the overall free energy change is 0. Now here we have the graphic representation that embodies the discussion from the previous slide. Again, to lengthen the glycogen chains, glucosyl residues are added from UDP glucose to the non-reducing ends of the chain by glycogen synthase. The anomeric carbon of each glucosyl residue added on to the growing glycogen molecule is attached in an alpha 1,4 glycosidic bond to the hydroxyl group on carbon 4 of the terminal glucosyl residue of the glycogen molecule. Now, this reaction goes on until the growing chain is 8 to 10 glucosyl units long. Now, when a chain has grown long enough, that is, if it is already 8 to 10 glucosyl units long, a different enzyme called 4,6-transferase, also known as glycogen branching enzyme, cleaves a part of it and transfers and attaches it to another site on the glycogen molecule via an alpha 1,6 glycosidic bond. There is now a new branch besides the original chain from which it was cleaved. Now, both chains then continue to lengthen until they are both long enough to produce two new branches. So we are now drawing near the end of the glycogenesis discussion. But there is one more member of this pathway that has not yet been discussed at length, the glycogen primer. Now, one thing about glycogen synthase is that it elongates the chains of a pre-existing glycogen molecule. But how does a glycogen molecule materialize in the first place? It begins with the production of a protein called glycogenin. It has the capability of autoglycosylation, wherein it attaches the glucosyl residue coming from UTP glucose to the hydroxyl group of a tyrosine, or sometimes a serine residue on its own structure. Now, glycogenin then catalyzes the transfer of a further 7 glycosyl residues, each linked to each other via alpha 1,4 glycosidic bonds to form the glycogen primer. In summary, the product of glycogenesis is a highly branched glycogen molecule. As mentioned earlier, the branching of glycogen serves two major roles: to increase the number of sites for synthesis and degradation, and to enhance the solubility of the molecule. Glycogen synthase is the regulated enzyme in this anabolic pathway. In the previous video, we covered glycogenesis. In this video, we will tackle the functional opposite of the glycogenesis pathway, glycogenolysis. Now, glycogenolysis basically involves phosphorylases reactions. Phosphorylases is the breakdown of a bond using a phosphate ion as a nucleophile. Now, the enzymes catalyzing such reactions are collectively termed as phosphorylases. The substrate is usually included in the name of the enzyme. Now, the two enzymes involved in glycogenolysis are glycogen phosphorylase and the debrancher enzyme. So, in glycogenolysis, glycogen phosphorylase gains access to the non-reducing ends of the branches of glycogen. From there, it successively cleaves glucosyl residues by adding phosphate to the anomeric carbon of the terminal glycosidic bond. A molecule of glucose 1-phosphate is released, and there is now a new free hydroxyl group on the carbon 4 position of the new terminal glucose residue. Glycogen phosphorylase has a limitation, however. We let each of the blue structures here represent one glycogen phosphorylase enzyme. As long as the chain is more than 4 glucosyl residues long, it performs its function just fine. The problem occurs when only 4 glucosyl residues are left in the branch or branches. The enzyme cannot act on the glycosidic bonds of these 4 glucosyl residues closest to the branch point. This is because the branching chain sterically hinders a proper fit into the catalytic site of the enzyme. Where glycogen phosphorylase falls short, the debrancher or debranching enzyme comes to the rescue. The debrancher enzyme, sometimes called 4,4-transferase, is responsible for removing the 4 glucosyl residues closest to the branch point. It has two catalytic activities, a transferase activity and a glucosidase activity. Now, their functions will be demonstrated in the slides that follow. The transferase activity of the debrancher enzyme removes a unit containing 3 glucosyl residues and adds it to the end of a longer chain by an alpha 1,4 glycosidic bond, hence the name 4,4-transferase. Now, all that remains of the branch is a single glucosyl residue attached to the branching point via an alpha-1,6 glycosidic bond. On the other hand, the glucosidase activity, or more specifically, the alpha-1,6 glucosidase activity of the debrancher enzyme, is the one responsible for removing this remaining glucosyl residue. Now, once that is taken care of, degradation of glycogen by glycogen phosphorylase resumes. And finally, some amount of glycogenolysis also occurs within the lysosomes of cells. This occurs when small glycogen particles become surrounded by membranes that then fuse with the lysosomal membranes. A lysosomal glucosidase is the one responsible for hydrolyzing this engulfed glycogen to glucose. This is the last of this 4-video series on glycogen metabolism. So this video will be noticeably longer than the first 3 because we'll be discussing how both arms of glycogen metabolism are regulated. The material can get confusing if you miss some bits of detail during the course of the discussion, so I highly recommend that you listen closely. Now, additionally, amongst other enzymes, we will be revisiting the phosphorylation cascade that I mentioned in passing in one of the previous videos. Now, this cascade consists of protein kinase A, phosphorylase kinase, and glycogen phosphorylase. The reason for this is that these are among the key players in the regulation of both glycogenesis and glycogenolysis. Now, as a review, kinases act by covalently attaching a phosphate group to its substrate, with that phosphate group coming from ATP, as is humorously depicted in this cartoon. Phosphorylation of an enzyme is often associated with its activation, but a lot of times, phosphorylation also results in deactivation. Though not represented here, the functional opposite of kinases are phosphatases, which remove the phosphate group initially attached by kinases. As you will see eventually, protein phosphatases are enzymes that counter the effects of protein kinase A. Now, one thing to understand about regulation of glycogen metabolism is that regulation matches the function of glycogen in the tissues involved. In the liver, glycogen metabolism is regulated by changes in the insulin to glucagon ratio, changes in blood glucose levels, and the catecholamine epinephrine. In skeletal muscle, on the other hand, levels of AMP, calcium ions, which are released during muscle contraction, and epinephrine regulate glycogen metabolism. Of note, skeletal muscle is insensitive to insulin. The principal enzymes in glycogen metabolism that are subject to regulation are glycogen phosphorylase and glycogen synthase. They are regulated in opposite directions by either allostatic mechanisms or covalent modification by reversible phosphorylation. You'll be able to appreciate this more later. Glycogenolysis and gluconeogenesis are activated together by the same regulatory mechanisms. However, relative to gluconeogenesis, glycogenolysis responds more rapidly with a greater outpouring of glucose to match the demand. So essentially, our liver glycogen stores are therefore a rapidly rebuilt and degraded store of glucose, ever so responsive to small and rapid changes in blood glucose levels. Now let's pay attention to this table here. This table showcases effects of fasting on liver glycogen content in humans, and how glycogenolysis responds more rapidly compared to gluconeogenesis. During up until 24 hours of fasting, you can see that the rate of glycogenolysis is fairly constant. However, in a prolonged fast, the rate of glycogenolysis decreases significantly as the liver glycogen supplies dwindle. Now, in order to understand better how regulation of glycogen metabolism is carried out, a good distinction between the active and inactive forms of glycogen phosphorylase and glycogen synthase has to be made. A particular nomenclature is assigned depending on the enzyme's state of activity and phosphorylation. The active form of glycogen phosphorylase is phosphorylated and is assigned the nomenclature A. Conversely, its inactive form is dephosphorylated and is termed glycogen phosphorylase B. On the other hand, the active form of glycogen synthase is dephosphorylated and is assigned the nomenclature I or A, while its inactive form is phosphorylated and is termed D or B. Of note, phosphorylation of glycogen synthase is far more complex than glycogen phosphorylase because the former has multiple phosphorylation sites and is acted on by up to 10 different protein kinases. Glycogen phosphorylase, on the other hand, has only one serine per subunit that can be phosphorylated. Phosphorylation by protein kinase A, as we shall see later, facilitates the subsequent addition of phosphate groups by other kinases, something that is called hierarchical or synergistic phosphorylation. On the other hand, glycogen phosphorylase, which has only one serine per subunit, can be phosphorylated by only phosphorylase kinase. Let us now take a metabolic scenario wherein glucagon levels are increased while insulin levels are decreased. When does this particular scenario occur? And which signaling pathway is activated by the predominant hormone glucagon? The answers to these questions are 1. During the fasting state, and 2. Glucagon makes use of the adenylyl or adenylate cyclase pathway by binding to and activating a G protein coupled receptor or GPCR. Now recall that binding of glucagon to the GPCR causes the G protein associated with it to dissociate into its alpha and beta gamma subunits, with the former activating adenylyl or adenylate cyclase, which then synthesizes cyclic AMP or cAMP from ATP. This cAMP then binds to the inactive form of protein kinase A or pKa for short, resulting in the activation of its catalytic subunits. pKa activates phosphorylase kinase by phosphorylation. The activated phosphorylase kinase then phosphorylates inactive glycogen phosphorylase, particularly at its specific serine residues, thereby activating it. And as you can recall from glycogenolysis, glycogen phosphorylase breaks down glycogen into its glucose 1-phosphate substituents, with glucose 1-phosphate eventually getting converted to glucose 6-phosphate. In the liver, glucose 6-phosphate loses its phosphate group because of glucose 6-phosphatase. The free glucose now leaves the liver and goes into the bloodstream to contribute to increasing blood glucose levels, which is the ultimate end effect of glucagon. And because during fasting, when glycogenolysis is activated, it is only logical that the process of glycogenesis is switched off. How does this happen? Let's go back to pKa from earlier. While phosphorylating phosphorylase kinase, pKa also adds a deactivating phosphate group to glycogen synthase, thereby decreasing its activity. This essentially prevents the synthesis of glycogen when in fact glycogen breakdown is needed more to maintain blood glucose levels because the individual is in the fasting state. Now notice that the solid black arrows or lines denote the reactions in glycogenolysis, which are activated during the fasting state. On the other hand, the red dashed arrows or lines denote reactions in glycogenesis, the activities of which are decreased in the livers of fasting individuals. As one shifts from the fasting state to the fed state following a meal, enzymes called protein phosphatases become upregulated, reversing the effects of glycogenolysis. Now, these enzymes remove the serine-bound phosphate groups that were initially attached by the kinases, responsible for adding them during glycogenolysis. The result is activation of glycogenesis and inhibition of glycogenolysis. Basically, these protein phosphatases were previously inhibited during the fasting state, at the same time that pKa and phosphorylase kinase are adding phosphate groups to enzymes. Hepatic protein phosphatase 1, or PP1 for short, is one of the major protein phosphatases involved in glycogen metabolism. It removes phosphate groups from phosphorylase kinase, glycogen phosphorylase, and glycogen synthase. In the fed state, we expect the insulin to glucagon ratio to be increased. These elevated levels of insulin indirectly activate hepatic protein phosphatase 1. Insulin makes use of the tyrosine kinase pathway to effect this activation. Now, the reason why it was mentioned that insulin indirectly activates hepatic protein phosphatase 1 is because it exerts its effects on this enzyme by way of another intervening enzyme called the insulin-sensitive protein kinase, which is part of the tyrosine kinase signaling pathway that insulin has initially activated. The activated insulin-sensitive protein kinase then phosphorylates hepatic protein phosphatase 1 to activate it. Again, note what was discussed in an earlier video about the enzymes being in close proximity to their substrate glycogen. Now, by activating hepatic protein phosphatase 1, glycogen synthase gets dephosphorylated and activated. Phosphorylase kinase also gets dephosphorylated, but it gets deactivated in the process. The end result is activation of glycogenesis. Now, this figure here is from Harper's and is similar to the figure from 3 slides ago, except that here it focuses only on the control of glycogen phosphorylase, as well as how it relates with phosphorylase kinase and protein kinase A, which is termed here CAMP-dependent protein kinase. Take note from earlier that insulin activates protein phosphatase 1. Activated pKa during glycogenolysis, on the other hand, inhibits protein phosphatase 1 indirectly by phosphorylating and activating a protein called inhibitor 1, which in turn inhibits protein phosphatase 1. Overall, the result of glucagon influence on hepatocytes is activation of glycogen phosphorylase with resultant glycogenolysis. Now, this is a part that involves glycogen synthase, which is deactivated in the fasting state. Again, you have protein kinase A, or CAMP-dependent protein kinase, as what is being called in this figure, phosphorylating and inhibiting glycogen synthase. As with previous, you also see here protein kinase A activating inhibitor 1, which in turn inhibits protein phosphatase 1, further inhibiting glycogenesis. Now in this figure, we integrate all enzymes and show how each relates to each other. This one now looks a lot similar to the original figure I presented earlier on. The content being conveyed is the same nonetheless. Now, going back to insulin. Insulin is the principal regulator of glycogen synthesis and degradation. Blood insulin levels change to a greater degree with the feeding-fasting cycle than do blood glucagon levels. Now, besides making use of the tyrosine kinase pathway, insulin is likewise able to affect the expression of GLUT4 transporters on hepatic cell surfaces and activation of glycogen synthase through activation of another signaling pathway, the PI3K-AKT pathway we shall be discussing next. Now here is the PI3K-AKT pathway. As you can hopefully recall from the signal transduction topic, the tyrosine kinase receptor for insulin activates the RAS-MAPK pathway, which is perhaps responsible for many of the anabolic effects of insulin. Now, the signaling pathway branches at IRS1, wherein it activates a protein called phosphatidylinositol 3 kinase, or PI3 kinase. This in turn phosphorylates phosphatidylinositol 4,5 bisphosphate, or PIP2, to phosphatidylinositol 3,4,5 triphosphate, or PIP3. By the way, PTEN, which stands for phosphatase and tensin homolog, is a PIP3 specific phosphatase that converts it back to PIP2. As you will learn eventually under the biochemistry of cancer, PTEN is encoded by a tumor suppressor gene in humans. Now, PIP3 then goes on to activate a protein kinase called protein kinase B, or PKB for short. Now, PKB triggers the movement of glucose transporter GLUT4 to the plasma membrane in hepatocytes, facilitating internalization of extracellular glucose. Likewise, PKB phosphorylates and inactivates another kinase called glycogen synthase kinase 3, or GSK3. In its non-phosphorylated form, GSK3 is active and phosphorylates glycogen synthase to inactivate it. Now, with GSK3 phosphorylated and deactivated, glycogen synthase remains activated to synthesize glycogen using the glucose molecules internalized by the GLUT4 transporters. Now, activated hepatic glycogen phosphorylase has 3 known allosteric inhibitors, namely ATP, glucose-6-phosphate, and glucose. Now let's inject some rationalization into the allosteric inhibition of glycogen phosphorylase by ATP, glucose-6-phosphate, and glucose. When there's already enough ATP produced, there's no further need for more ATP production from glucose metabolism for the time being. So at that point, there will be no need for glycogen phosphorylase to cleave off more glucosyl units from glycogen to produce ATP. Similarly, when there's already enough glucose-6-phosphate and free glucose molecules that can be channeled to energy-producing metabolic pathways, there is no further need for glycogen phosphorylase to cleave more glucosyl units from glycogen for the moment. We now leave glycogen metabolism regulation in the liver and move on to glycogen metabolism regulation in the skeletal muscle. Now, skeletal muscle glycogen is degraded only when the demand for ATP generation is high. This happens during exercise. But unlike the liver, there is no glucose 6-phosphatase in skeletal muscle. So the glucose 1-phosphate that is produced with glycogen breakdown does not get dephosphorylated. Instead, it is converted to glucose 6-phosphate and can either enter glycolysis, which is most likely going to occur during exercise, the pentose phosphate pathway, or other metabolic pathways. Now, as a general rule, anaerobic glycolysis occurs in tissues that have fewer mitochondria, by that we're referring to RBCs, as well as in tissues with a higher content of glycolytic enzymes and higher levels of glycogen or fast-twitch glycolytic fibers, and by that we're referring to skeletal muscle. Okay, now this one we already know. As a consequence of this lack of glucose 6-phosphatase, glucose is no longer a physiologic inhibitor of activated glycogen phosphorylase in skeletal muscle, unlike in the liver. Glycogen, however, exerts a stronger feedback inhibition on skeletal muscle glycogen synthase. This is due to the fact that there is a smaller amount of stored glycogen per gram weight of muscle tissue compared to liver. Now, recall from earlier how glucose 1-phosphate produced during glycogenolysis has to be converted to glucose 6-phosphate, so it can either enter glycolysis or other metabolic pathways. Now, that confirms the fact that in skeletal muscle, both glycogenolysis and glycolysis can be activated together. Now, besides being insensitive to insulin, skeletal muscle is also insensitive to glucagon. Now, levels of cyclic AMP are increased instead by the action of epinephrine or norepinephrine for that matter. Now, from basic physiology, we already know that these catecholamines are secreted as part of the fight or flight response. Now, adenosine monophosphate or AMP is an allostatic activator of muscle glycogen phosphorylase. Now, recall from bioenergetics that ADP or alpha delta papa that is produced from the cleavage of ATP or alpha tango papa still has a high energy phosphate bond left. And in many instances of high energy demand, ADP gets further cleaved into AMP and inorganic phosphate. Thus, a rise in AMP levels is reflective of dwindling ATP levels. Thus, it makes biological sense that AMP activates muscle glycogen phosphorylase to cleave more glucosyl units or glucosyl residues from glycogen to be used for energy production. Now, let's go back to the same illustration as earlier. Epinephrine with skeletal muscle acts very much the same way as glucagon with hepatocytes. In skeletal muscle, however, epinephrine is able to make use of two types of adrenergic receptors, which are both GPCRs. The first one is a beta adrenergic receptor, which makes use of the adenylate cyclase pathway that you see here. The other receptor is an alpha adrenergic receptor, which makes use of the phospholipase C or PLC pathway. Now, as a review, phospholipase C is the effector enzyme in this pathway, and it effects the cleavage of PIP2 into second messengers IP3 and diacylglycerol. IP3, being the soluble second messenger, diffuses into the cytoplasm and is believed to activate and open calcium channels on the endoplasmic reticulum, resulting in a flux of calcium ions. The release of calcium from the sarcoplasmic reticulum in skeletal muscle, however, is more a result of neural stimulation than epinephrine-stimulated uptake. Now, these calcium ions go on to activate a protein called calmodulin, which is closely associated with a phosphorylase kinase. This activated complex now goes on to phosphorylate and activate target phosphorylase enzymes. Now, how do the calmodulin and its associated kinase relate to glycogen metabolism? In glycogen metabolism, the kinase closely associated with calmodulin is called the calmodulin-dependent kinase. Through epinephrine, the complex is activated, which in turn phosphorylates glycogen synthase, making it less active, thus making glycogenesis less likely to push through. So, as was recently mentioned, the release of calcium ions from the sarcoplasmic reticulum in skeletal muscle is more a result of neural stimulation than epinephrine-stimulated uptake. And just to reiterate, The effects of epinephrine-stimulated phosphorylation by pKa on synthesis and degradation of skeletal muscle glycogen are similar to those occurring in the liver. As you may have already observed at this point, one important difference between skeletal muscle and hepatic glycogen metabolism lies with phosphorylase kinase activation, as will be discussed further in the next couple of slides. Now, in skeletal muscle, there should both be protein kinase A activation by epinephrine and calcium release from the sarcoplasmic reticulum, secondary to nerve impulse and muscle contraction, in order for skeletal muscle phosphorylase kinase to be fully activated. Now, as was also discussed earlier on, calcium levels increase also because epinephrine stimulates the alpha-adrenergic receptors that make use of the phospholipase C pathway. Again, calcium ions bind to calmodulin, which then associates as a subunit with phosphorylase kinase, thus partially activating it. Recall as well that protein kinase A is also needed for full activation of phosphorylase kinase. Additionally, AMP is an allosteric activator of skeletal muscle glycogen phosphorylase. Now, let me end this section on regulation by showing you a graphic summary of the allosteric regulators of the two main enzymes in glycogen metabolism, glycogen phosphorylase and glycogen synthase, in both the liver and skeletal muscle. What is not included here is glycogen as a strong feedback inhibitor of muscle glycogen synthase. Now, I'll just be breezing through glycogen storage diseases. I'll leave this topic for you to read on your own. So basically, glycogen storage diseases are a group of inherited disorders characterized by deficient mobilization of glycogen. Now, this often leads to deposition of abnormal forms of glycogen. Now, glycogen storage diseases generally result in liver damage and muscle weakness, with some even resulting in early death. The resulting organ damage is frequently due to deposition of abnormal forms of glycogen in those organs. Now, this table here, which I lifted directly from Harper's, summarizes the different glycogen storage diseases along with the specific enzyme or enzymes affected. Again, as I said, I will leave this topic for you to read on your own. The key here is that besides knowing what enzyme or enzymes are affected in each type of glycogen storage disease, understand as well what the function of each enzyme is, and visualize the biochemical consequences when they are inactivated or deficient. That is, what substrates would accumulate, etc. And try to correlate this with the clinical features listed in this table.